Assalamualaikum and good morning everyone. My name is Muhammad Hanif Afifi bin Nur Yusman from the Bachelor of Science with Honour in Applied Microbiology program. Today I will be presenting my final year project research proposal entitled Isolation and Characterization of Drought-Tolerant Bacteria Capable of Producing Gibberellins from Soil in Jelebu District. First, I will briefly explain the background of this research. Agriculture plays a critical role in Malaysia's economy. However, in recent years, agricultural productivity has been increasingly affected by climate variability, especially prolonged drought events. Studies have shown that drought stress significantly reduces crop growth and yield, particularly in crops that are sensitive to water availability. The Jelebu district in Negeri Sembilan is of particular interest because it is located within the Titiwangsa mountain range, resulting in low annual rainfall, making it the driest area in Peninsular Malaysia. Despite these harsh environmental conditions, vegetation in Jelebu continues to survive. This suggests that natural biological support system, particularly drought-tolerant plant growth-promoting bacteria, may play an important role in helping plants to survive. This brings us to the main problem that this study aims to address. Although drought has become a growing threat to Malaysian agriculture, current mitigation strategies still rely heavily on chemical fertilizers and synthetic plant growth regulators. These approaches are often costly, environmentally damaging, and unsustainable for long-term use. While beneficial bacteria capable of producing plant hormones such as gibberellin have been studied intensively, most existing research focuses on foreign or temperate region strains. These strains often perform poorly when introduced into Malaysian soils due to differences in soil acidity, nutrient availability, and temperature. Currently, there is limited scientific data on native drought-tolerant gibberellin-producing bacteria from Jelebu soils, which restrict the development of locally adapted and sustainable biological solutions. Based on this problem, this study is guided by two main research questions. First, which culturable bacteria from Jelebu soils are drought-tolerant and capable of producing gibberellins? Second, what are the morphological and biochemical characteristics of high-performing isolates? To answer this question, this study has two main objectives. The first objective is to isolate culturable bacteria from representative soil samples in Jelebu and screen them from drought-tolerant using PEG-induced osmotic stress, as well as preliminary gibberellin production using a colorimetric assay. The second objective is to characterize selected high gibberellin producing isolates based on colony morphology, microscopic features, and basic biochemical tests. The significance of this study lies in its potential to bridge microbial ecology with practical climate adaptation strategies in Malaysian agriculture. By isolating bacteria from Jelebu, this research focuses on a naturally selected gene pool that has already adapted to prolonged drought conditions. These native bacteria are more likely to survive and function effectively when reintroduced into local soils compared to imported commercial strains. Additionally, identifying bacteria capable of producing exogenous gibberellins provides a biological mechanism to maintain plant growth during drought when plants naturally suppress hormone production. This approach reduces reliance on synthetic chemicals, supports long-term soil health, and contributes toward climate resilient and sustainable agriculture practices. Several studies have shown that drought affects both soil health and plant development. The depletion of soil moisture leads to soil particles to come into closer contact, reducing the volume of pore spaces and subsequently limiting oxygen availability for soil-dwelling organisms. Moreover, water stress causes plant stunting, reduced photosynthesis, oxidative damage, and nutrient deficiency. Next, based on previous research, the interaction between drought-tolerant bacteria with plant is that the bacteria will produce exopolysaccharides that form protective biofilms around roots, improving moisture retention in the rhizosphere. It also produces osmoprotectants and induces systemic tolerance by activating plant antioxidant defenses, thereby reducing oxidative damage. Next, gibberellin's function in plant development is that it regulates processes such as seed germination, stem elongation, and leaf expansion by promoting cell division and loosening the cell wall. By introducing gibberellins producing bacteria, it can restore hormonal balance, maintain growth, and prevent permanent dormancy. Malaysian soils, especially in Jelebu, remain underexplored and foreign strains often fail due to poor ecological fitness. This highlights the need for isolating and characterizing native gibberellin-producing bacteria for developing sustainable biofertilizer to enhance plant growth. This study follows a laboratory-based experimental design. First, approximately 100 grams of rhizosphere soil samples will be collected from Jelebu at a depth of 5 to 15 centimeters, stored at 4 degrees Celsius and processed within 24 hours. Next, all media and solution including nutrient agar, nutrient broth, saline solution polyethylene glycol PEG 600, supplemented broth and sucrose amended agar is to be prepared. Soil bacteria will be detached by suspending 10 grams of soil in 90 ml of sterile 0.85% saline, shaking at 150 rpm for 30 minutes, followed by tenfold cereal dilution from 10 to the power of -1 to 10 to the power of -6 with vortex mixing to obtain suitable concentration for colony isolation. Next, bacteria will be isolated from soil using cereal dilutions from 10 to the power of -1 to 10 to the power of -6 and spread plating on nutrient agar, followed by repeated quadrant streaking to obtain pure cultures. The plates are incubated at 30 degrees Celsius for 24 to 48 hours. After incubation, the number of colonies is counted. Next, to screen for drought tolerance, isolates will be cultured in nutrient broth supplemented with PEG 6000 at osmotic potential of -0.4, -1.0 and -1.5 megapascal, and growth will be measured using OD 600 readings. Moving on, drought-tolerant isolates will then be screened for gibberellin production. Bacterial isolates are inoculated into 50 ml of nutrient broth and incubated at 30 degrees Celsius for 7 days on a rotary shaker at 150 rpm. Cultures are centrifuged at 10,000 rpm for 15 minutes, then acidified to pH 2.5. Methanol extract will react with reagent for color development, and absorbance is measured at 254 nanometer. Next, selected high-performing isolates will be characterized based on colony morphology, Gram-Staining reaction, and functional biochemical tests including catalase, oxidase, and isopolysaccharide production. Lastly, for data analysis, all experiments are conducted in triplicate, with growth and gibberellin production data analyzed using one-way ANOVA followed by 2 KISS post-hoc tests to identify statistically significant differences among isolates. The expected outcome of this study is the successful identification and characterization of native drought-tolerant gibberellin-producing bacterial isolates from Jelebu soils. These findings will contribute baseline scientific data for future greenhouse or field studies and support the development of bio-based drought mitigation strategies. Potential challenges include time limitation, laboratory safety risks, and equipment availability. To address this, high-performing isolates will be prioritized, strict lab protocols will be followed, and experiment will be carefully scheduled.